we are integrating advanced machine learning with patient-derived midbrain organoids to discover disease-modifying therapies for Parkinson's disease, the second most common neurodegenerative disorder. Parkinson's disease is a progressive terminal neurodegenerative disorder characterized by the degeneration of midbrain dopamine neurons, affecting approximately 10 million people worldwide. Despite substantial investments in research, there are no approved disease-modifying therapies that slow or halt its progression due to the lack of predictive translational models for drug discovery. Existing treatments primarily provide symptomatic relief. Our patient-derived midbrain organoids accurately simulate dopamine neuron degeneration, helping to fill this critical gap. Using GBA1 mutant iPSCs from Parkinsonian patients, we developed organoids that exhibit progressive dopamine neuron loss. RNA-seq analysis identified critical dysregulated processes, including axon guidance and neurotransmitter secretion. Our platform overcomes the limitations of traditional gene set enrichment methods by providing more precise insights into PD progression. We developed a foundation model-based network analysis workflow utilizing single-cell foundation models like SCGPT and GeneFormer within the NVIDIA BioNeMo framework. By fine-tuning the model with midbrain organoid data, we created a network that captures Parkinson's disease-specific biological processes, enhancing target discovery and translational research. The workflow includes dimension reduction, community detection, and cluster annotation, allowing us to identify disease-specific clusters linked to known biological processes. This established network enables the exploration of gene expression studies, enhancing our understanding of the disease model and drug mechanisms. We've created an interactive website to analyze the organoid network, allowing users to evaluate gene clusters and disease mechanisms. Different nodes represent genes, and they cluster by similar expression profiles. For example, red nodes indicate upregulated genes, while blue nodes show downregulated genes, reflecting changes consistent with dopamine neuron loss and neural precursor function. Our rigorous benchmarking validated our approach by comparing various foundation models, including SCGPT and GeneFormer, and traditional gene clustering algorithm to assess their performance in capturing PD-relevant pathways. The fine-tuned SCGPT model outperformed others in detecting processes critical to PD pathology, such as neurogenesis and cell cycle dysregulation. Our patient-derived midbrain organoid models captured key pathogenic pathways in PD, especially in dopamine neurons. Bulk and single-cell RNA-seq revealed consistent downregulation of genes involved in neurogenesis and cell cycle differentiation, aligning with that observed in PD tissue. Additionally, bulk RNA-seq showed cell cycle dysfunction, likely from neural precursors. Cell sequencing shows consistent results in neurogenesis and cell differentiation seen in both patient-derived brain organoids. It also reflects genotype-specific pathogenicity, such as lipid metabolism dysfunction in the GBA1 organoid, but oxidative phosphorylation in the LRRK2 organoid. This highlights the platform's potential as a clinical trial in a dish for Parkinson's research. In conclusion, our foundation model-based network analysis utilizing midbrain organoids has successfully captured key biological processes involved in PD progression. The SCGPT fine-tuned model offers new opportunities for drug target discovery and biomarker identification. Future steps include using this platform for in silico perturbation screening to simulate drug responses and identify potential treatments. We also aim to integrate models like the NVIDIA BioNeMo version 2 and expand our analysis to other neuropsychiatric and neurodevelopmental disorders, transforming drug discovery in these fields. During the hackathon, we formed a collaborative team, including members from Brainstorm Therapeutics, alongside experts like Birkin and Anna in machine learning and data processing, and Jens, who provided organoid data sets. Thank you for your attention, and we look forward to any questions you may have.